This last section is on molecular defenses. I think it's a little harder to relate to. We've all experienced inflammation and fever, but we don't always think about our body fighting these pathogens on a molecular level. So with this section, we'll talk a little bit more about complement that we'll see in the inflammatory process. We'll also be seeing complement again in the adapted or acquired immune system. And then we'll also talk about interferon. So there's two types of interferon. I'm mainly going to be talking about type 1. I often think that interferon interferes with viral infections. And so we're mainly going to talk about type 1. So it's a little easier to talk from a picture. So I'm going to go to the next frame where we can actually see interferon in action. So let me get my pen out so that I can draw on this picture. So over here, we've got our first cell. So this is a cell that has been infected by a virus. Remember, viruses, they have to attach to a cell. They have to adsorb, and then they either have to get into the cell or they have to inject their DNA. So you can see with this cell, it's being overrun by a virus. So that virus is getting in, taking over the host cell machinery and making its parts. Well, as it takes over that host cell machinery, it actually activates the gene for interferon. Now that cell starts to make interferon, and interferon is a protein that then travels next door to the neighboring protein to signal to that protein that, okay, there's trouble in the area and you better get ready to fight. Now, this first cell, this is going to be a goner because the virus has gotten in and it's going to take over, and then that cell is going to lyse. This is kind of like a warning sign to the neighbors. So this cell is a goner. It's like, save yourself, but there is trouble afoot. So when that interferon binds to the second cell, what it does is it prompts the cell to make these antiviral proteins. So up here with this picture, when it's making those antiviral proteins, when that virus adsorbs, tries to take over the host cell machinery, those antiviral proteins actually block that viral replication. Specifically, they interfere with how that virus can make its nucleic acid. And so that virus is not going to be able to continue to replicate. So in this case, it would be an abortive viral infection. The first cell is a goner. This is kind of like a warning sign to the neighborhood that trouble is coming. So that is type 1 interferon. Now, for people that are fighting chronic illnesses and viruses, so something like genital warts or hepatitis C, they can actually administer recombinant interferon. And so this helps those white blood cells work better, helps to boost the immune system so the person can actually fight that virus. Now, we know that anytime that you are giving medicines, there could be some secondary side effects. Recombinant interferon is not that stable, so it tends to break down. But they have found that with some success in treating these chronic viral infections. We've already mentioned complement, and complement are basically 20 different proteins. So I'm not going to ask you to memorize all of these complement proteins, but we will talk a little bit about what complement does. So complement can attract those phagocytes to the scene. So remember those white blood cells that are going to run into the face of danger. They can also enhance phagocytosis by opsonization. So remember that term opsonization means that they're going to coat something so that those phagocytes can actually bind to that pathogen. They can also poke holes in the membranes of bacteria, and they can regulate some inflammatory effects. So right here are the three main effects of complement. Number one is that coating, opsonization. So coating the surface of the infectious agents so that antibodies bind to it, and then phagocytes can actually bind to that pathogen and gobble it up. We also see different complement in the inflammatory process. So it increases chemotaxis of phagocytes. So those immune cells are going to be running into the face of danger. Also increases the release of histamine. Remember, histamine is that flare gun going off. So that's going to allow those phagocytes to move into the area. And there are also certain complements that can form a membrane attack complex. And basically what this does is it pokes holes into the bacterial cell wall. So if you poke a hole in a bacteria, bye-bye bacteria. It's not going to be able to live. Obviously, this is not going to attack our cells, only those bacterial cells. The last thing that we will talk about are acute phase proteins. So cytokines, remember, those are the immune chemicals that these blood cells, these white blood cells, can communicate with. They can release these cytokines like interleukin 6 that will travel to the liver and induce the liver to make these acute phase proteins. So what are these acute phase proteins? Well, a lot of them can bind and opsonize, remember, coat infectious agents, and also activate complement. So again, kind of bringing in the cavalry and trying to get rid of these things. The liver secretes C-reactive protein. This is actually in response to inflammation that can activate complement. This is something that clinicians could measure to find out if there is inflammation in the body. So if those C-reactive protein measurements are up, that means that the patient probably has a lot of inflammation. I mentioned that my husband has Crohn's, and this is one of the measurements that they use to find out if his immune system is basically causing inflammation and attacking his intestines. So if his C-reactive proteins are really high, that means there's a lot of inflammation. If it's lower, and I don't think it's ever been normal since he's been diagnosed with Crohn's, but if it is lower, that means that his immunosuppressant is actually working. There's another protein, mannose binding protein. This binds to mannose, which is a sugar in many bacterial and fungal cell membranes. We don't have this sugar. And so what this protein does is that it sticks to it, binds to it, flags it, marks it, and tags it so that these phagocytes can actually find it. So these are two specific acute phase reactants, C-reactive proteins and mannose binding protein.